Hello, Biology 230. This is Dr. Chastity Bradford, and I will begin Unit 2, Chapter 7.1 in Cell and Genetic Biology 230 here at Tuskegee University, and we will cover membrane structure and function. And I believe this chapter in this unit is one of the most important foundational chapters in this course. Let's begin to discuss the plasma membrane and why is it important. It's called the edge of life, because it's the boundary that separates the living cells from their surroundings. What do we know about it? We know that this membrane is highly selective about what can cross it. So it's selectively permeable, okay? The plasma membrane is a selectively permeable membrane. And throughout this entire chapter, you're going to learn how membranes control what passes across the membrane. What is a membrane? What's in a membrane? These are important questions to ask, and the answers you will receive throughout this lecture. Take a look at this ancient membrane. What does it really remind you of? This is something that ancient Egyptians used. They used it for protection from the sun. So the men would cover their penises with this sheath, often made of linen. And throughout the years, we know now that it's called a condom. We know of it now as a condom. And so even today, it's used for protection, not necessarily from the sun, but it's used for protection from sexually transmitted diseases. And so, in an essence, this is a membrane, right? Condoms are membranes, and they are selectively permeable. And they were designed to prevent the permeability of certain viruses. And more now, you hear in the news about the importance of using protection like a condom, especially for the prevention of sexually transmitted diseases such as HIV. So as you think about membranes, cell membranes, I want you to really think about the life application of membranes, because cells really must regulate what comes in and out of them in order to survive. Just like cells do, we as individuals have to regulate what we're exposed to in order to survive. The cell membrane consists of a number of proteins, a number of macromolecules. It's composed of cholesterol, phospholipids, sphingolipids, carbohydrates, and as I mentioned, proteins. Now, together with cholesterol and phospholipids, they form the lipid bilayer, which functions to be selectively permeable. It's a barrier, just like the condoms. It's a barrier between the cytosol, so that's inside the cell, and what's outside of the cell. The phospholipids, sphingolipids, carbohydrates, they come together to form glycolipids. Carbohydrates and proteins come together to form glycoproteins. And their functions include structural stability, cell-to-cell recognition, and immune responses. And in this lecture, this series of lectures, we will talk about the membrane and its function as a selective barrier. We've talked about in the previous chapter its function and structural stability. And in this chapter, we'll talk about its function in cell recognition and briefly mention its function in immune responses. Cellular membranes, they're fluid mosaics of lipids and proteins. They're fluid mosaics of lipids and proteins. So you should be able to fill in these blanks based on what you learned from previous lectures. Blank are the most abundant lipids in the plasma membrane. Blank are amphipathic molecules. What does amphipathic mean? They contain hydrophobic and hydrophilic regions. Amphipathic. And the fluid mosaic model states that a membrane is a fluid structure with a mosaic of various proteins embedded in it. You think of a mosaic, you think of a number of images, okay? Multiple images. So a membrane, this fluid mosaic model, talks about the membrane having a number of various proteins embedded in it, similar to a mosaic. So what is this lipid bilayer made of? Okay, if the external environment is water, what is this bilayer made of? I'm giving it away. The heads are made of something that you should know by now. Okay, and what you're seeing here is the image of these heads highlighted. Okay, and you see the tails. Okay, so it's a bilayer. One layer, the phospholipid heads. The second layer, of course, is composed of these hydrophobic regions, the fatty acid tails. Okay, hydrophilic head and hydrophobic tails. So this is a lipid, one layer, two layer, a lipid bilayer. Okay. What do we know about this fluid mosaic model, about this lipid bilayer model from the past? Okay. There are a lot of researchers that proposed a lot of different things about the plasma membrane and their thought about the model. And you know, as time progresses, as science progresses, as technology progresses, we learn more and more about different things. And so Davison and Danieli, in 1935, they proposed a model that looked like this. And what they proposed was that outside of the what they proposed is that outside of this bilayer, if you will, there were proteins on both sides, okay? And that the protein level, the protein layer, and the what we know now as the phospholipid heads, that this was the hydrophilic zone, okay? And that the hydrophobic zone was the inner component here, the fatty acid tails. So this was the Davison-Danieli proposed sandwich model. And in this model, as you see, the phospholipid bilayer lies between two layers of globular proteins. Okay? So these were some of the earlier images. Up until the 1960s, it was widely accepted as the structure of the membrane and also the structure of internal membranes. But after further investigation, it revealed a couple of problems. First, not all membranes are alike. Some differ in their thickness. Some, of course, when they stain them, some membranes appear stained. Some of the proteins were different. The percentages of proteins that were there were different, and membranes with different functions differed in their chemical composition and structure. So not all membranes were alike, and that's what they were proposing, that all membranes look like this, you know. And so it wasn't until 1972 that Singer and Nicholson proposed another model, the one that's more acceptable, that has been widely accepted and is still accepted today, and this is that fluid mosaic model. And I show you a few different versions of that model as it appears here. as it appears in B and as it appears here in C. So this is that phospholipid bilayer model. You see the two layers. And of course, they were proposing that the membrane proteins were dispersed throughout the entire phospholipid bilayer. So as you see here, all the proteins are throughout this lipid bilayer. Some of them are peripheral in their attachment. Some of them are transmembrane and they cross the entire membrane, as you see here, integral proteins. And so this is what they were proposing, that it's a mosaic of proteins dispersed within this bilayer. And the only regions that are exposed to the water were the hydrophilic regions. Okay? And so this is the widely accepted model today. And there, of course, the belief and the belief is that these hydrophobic regions, of course, are not in contact with the water, as opposed to the model previously. Let me show you the previous model. Okay, that is shown here. Okay. So, what else do we know about the fluidity of membranes? So, we know that membrane molecules are held in place by relatively weak hydrophobic interactions. And we know that these membranes are fluid. What does fluid mean? Just that they're not fixed. Okay? These membranes are not fixed. And you'll notice in one of the activities that we've asked you to do, it really gives a good visual depiction of the fluidity of membranes. And what you see here is a pictorial diagram that shows the plasma membrane here in orange. And then it kind of zooms in on this membrane and shows you the different layers and the different proteins that are associated with the plasma membrane. And then it zooms in even closer and it shows you the integral membrane proteins. It's one spans the entire lipid bilayer. Okay? And then it also shows you here, the alpha helical transmembrane segment, and now you should know that what level of protein structure this represents, this alpha helix represents. Okay. Of course, this over here just gives you a diagram that shows you the phospholipids are structured like this in their example, that the polypeptides are represented by strings here of amino acids. And you see, you should know what this functional group is by now. And that in green, you have carbohydrate chains, okay? And these are glycoproteins. So, the take-home message from this slide is that membranes are fluid. What do we really mean by that? Phospholipids within the membrane, they can move within that bilayer. So, it's not static. It's a fluid membrane, okay? And membranes must be fluid in order to work properly. And it's estimated that they're usually about as fluid as salad dressing. Okay? So really think about the membrane as a fluid model, okay? A fluid mosaic. And what would happen if our membranes, if our cell membranes were solid? What would happen? What would happen if our cell membranes were too fluid? If they were solid, what would happen? Think about this. If they were too fluid, what would happen? Okay? So, fluidity of membranes is extremely, extremely, extremely important to the survival of a cell. If a membrane is too solid, it changes the permeability of the membrane. And guess what? The enzymes within that membrane can't function. If a membrane was too fluid, then it couldn't support the membrane's function. Okay? This is how I want you to begin to think. Structure, function. So the temperature at which a membrane solidifies depends upon the components of that membrane. So once again, the components of the membrane are important and they dictate the fluidity of that membrane. So the type of lipids that are in the membrane and whether or not cholesterol is there or not influences a membrane's fluidity. And remember that only animal cells have cholesterol, okay? Only animal cells have cholesterol, and cholesterol acts as a temperature buffer, and we will explain that later. This lecture. So the hydrophobic region of the membrane is composed of fatty acid tails, right? Now, if these tails are saturated fats or unsaturated fats, then that will determine the fluidity of the membrane. Because if you recall, the saturated fats, they can stack on each other, right? You can put one next to the other, next to the other. And that's why, palmitic acid, that's why margarine, butter, the sticks of butter, the sticks of margarine, and also the lard they used to use back in the day, that's why they're in solid form, because they're composed of these saturated fats. Now, if you had a membrane composed only of unsaturated fats, it would be extremely fluid because there are kinks here, okay? There are kinks. There's the double bonds that form these kinks in these fatty acid tails. So they cannot stack on each other. So saturated saturated fats, think of lard. Unsaturated fats, they have kinks because of these double bonds that exist, that are here, that are not present in saturated fats. Now these kinks and this chain of fatty acids prevents them from sticking together, okay? And so when you think unsaturated fats, you think about your olive oil, you think about your salad dressing that has the oil, okay? That form. So at room temperature, unsaturated fatty acids, unsaturated fats are liquid. Room temperature, saturated fats are solid, okay? So at low temperatures, okay, so think about this. So if a temperature cools, as the temperature cools, membranes will begin to switch from a fluid state to a solid state, okay? They go from a fluid state to a solid state. We're not talking about membranes that have cholesterol as the animals do, okay? Let's just say we're talking about a plant cell in a low temperature. It will want to go from a fluid state to a solid state, okay? But is there something in these membranes that will allow the membrane to survive? There has to be a way. There are evolutionary adaptations of membrane composition that determine membrane fluidity. So in an environment like this is a beautiful picture, I think, here to your left. You would think it's really cold. So organisms have to adapt to survive the temperatures that are here versus temperatures that are here. And think about as seasons change, how do these plant cells survive? You have some evergreen trees that are always green. You have some winter wheat that will come up in the winter. How does this wheat survive the winter? If we know that in cool temperatures, as we mentioned before, if we know that in these cool temperatures, that a membrane will shift from a fluid to a solid state. But we spoke earlier about the fact that in a solid state, the enzymes don't want to function if they're frozen, solid. So the membranes have to adapt somehow. And how do they do that? They compensate, of course, because living things compensate. And their method of compensation is to change the lipid composition in their membranes. They change it to prevent solidification of that membrane. This is crucial, so important that you understand this concept. And how would they do that? They would change the composition by increasing the unsaturated fats. in their membranes. This is the method by which winter wheat survives the winter. Okay? This is an evolutionary adaptation of membrane composition that allows cells to survive when seasons change. So I want you to enjoy laughing at me as I have come up with this way that I hope helps you remember how unsaturated fats are and how saturated fats exist, okay? Unsaturated fats, they make very fluid membranes. Get it? So they're super kinky. Get it? Do you get it? So when there are kinks in the membrane, when there are kinks in the fat, not the membrane, when there are kinks in the fat, they make more fluid membranes, right? So think about it that way. Okay? Now, animal cells, how have they adapted to these changes in temperature? How do our cells adapt and adjust such that when it's cold, our membranes don't freeze, or when we get too hot, our membranes are not super fluid? So animal cells have cholesterol, okay? And I want you to remember that cholesterol acts as a temperature buffer for the membrane because it resists changes in membrane fluidity as temperature changes. Okay. Cholesterol reduces membrane fluidity at moderate temperatures by reducing phospholipid movement. And as you see here in this bilayer, I have circled the cholesterol. See it here? In blue and here. So these are the cholesterol. This is cholesterol in the membrane of animal cells. So that when there are temperature changes, okay, the cholesterol is here. And what does it do? It reduces the phospholipid movement. So regardless of whether it's too hot, it will prevent the membranes from becoming superfluid. If the environment is cold, it prevents the membranes from freezing or from solidifying. So this is the way that animal cells have, this is the way the animal cells have adapted, okay, to resist changes in membrane fluidity as temperature changes. Important concept for you to get. So cholesterol is a steroid, and it has different effects on membrane fluidity at different temperatures. So let's look at this again. So a low temperature, when it's cool, how does the membrane prevent solidification? How does it keep the fluidity of the membrane normal? So in an animal cell, you're going to have what? Cholesterol. So if your body temperature is high and it's hot, how does your membrane prevent superfluidity? How does it prevent this phospholipid bilayer from becoming too fluid, if you will? How does it keep the fluidity normal? The presence of cholesterol. Okay. So now this is something where I want you to think and post your answers on Blackboard for your respective professors. And you have archaea organisms in this domain have what type of membrane composition? What would you expect the membranes of organisms in, say, a geyser that can live in a geyser? Okay. In this habitat, what do you expect their membrane type, their membrane composition to look like? Okay. In terms of the fatty acid composition, would you even expect cholesterol to be there? Okay. These are the things you have to know. And then in another environment, such as Antarctica, what are the expectations for the fish that can survive in this and live in this and thrive in this environment, what type of membrane composition should exist there in order for them to survive? So in terms of fatty acid composition, what type should be there? And answer the question, do you think that cholesterol is in their membranes or not? And why? And why not? Okay? And so with this concept, hopefully you now understand that phospholipids are the most abundant lipid in the plasma membrane, that phospholipids are amphipathic molecules. They contain both hydrophobic and hydrophilic regions, and the fluid mosaic model that Singer and Nicholson presented, that was a revised model and one that's highly accepted now. It states that the membrane is a fluid structure with a mosaic of various proteins embedded in it. Okay? So now we're going to move on to talking about membranes that are mosaics and how and what they're composed of and how these mosaics affect structure and function.